So good morning. As Shane said, I work for the European Medicines Agency. For the ones of you who are not Europeans, it's the place where we authorize the new medicines for the European Union. So we follow all the process, basically, of the development of medicines until their authorization. And putting this experience I have with the European Medicines Agency together with my previous life in alpha-1 antitrypsin deficiency, I try to prepare a presentation for you that can be a mix of what are the problems in developing new treatments for rare diseases in general and in particular for alpha-1 antitrypsin deficiency, and also how does this work? How do we authorize the medicines? What are we looking at when we authorize the medicine? And how much and where do patients contribute to that? I also wanted to, by the way, to dedicate my talk to Maurizio Luisetti. I know you celebrated him yesterday. It was really a great man and a great source of inspiration, and we all miss him a lot. So, going into my talk, I have no conflicts of interest, and I will speak about where are the problems in developing medicines for rare diseases, like I said, which are the regulatory pathways, and then some conclusions and some possible actions. I try to come up with some actions that you can do also as patient groups. So, we know what is rare because we've been told this many, many times, but actually you might want to remember that this is a working definition. It doesn't, I mean, rare is something that is rare, it can be whatever, but this is a working definition that has been created for public health and healthcare purposes. So it can slightly vary in different types of the word, and actually some rare diseases, it has a threshold in Europe of not more than 5 in 10,000. So some rare diseases that are close to the threshold, if they get better cured, can get out of it and not be rare anymore. But I prepared this slide just to show you that actually to have a rare disease can be very different. You go from progeria that has 25 patients in the whole world to cystic fibrosis that have 40,000 in Europe. And actually, this makes a lot of difference in the way and the possibilities you have to develop a treatment and to test its efficacy. So, we are having quite a number of medicines for rare diseases because you can see that the United States created a legislation for fostering the development of medicines for rare diseases already in the 80s, and we in Europe created this later, but we have reached, we have designated in 2014 198 products, and we have designated up to date around 1,000 in total. But if we look at what we authorize as orphan medicines, so medicines for rare diseases, you can see that most of them are usually cancers. So this reflects the number of medicines we authorize for all conditions. And then for respiratory diseases, and alpha-1 antitrypsin would be classified under respiratory even when it's when there was a treatment for liver disease, it's only 5%. And among these 5%, how many are for rare lung diseases? There are actually not many. There are some rare lung diseases that have been very heavily covered by industry. The most famous one is pulmonary hypertension, for which there are really a lot of treatments. The same is cystic fibrosis, for which we have nowadays quite a number of treatments, including ivacaftor that I will mention later on in my presentation, which is actually a treatment that can change the natural history of the disease because it's really acting on the protein that has a defect in cystic fibrosis. We have more than 40 designations for cystic fibrosis, and look at this, we have only 4 for alpha-1 antitrypsin deficiency, and not even very recent actually, because this is 2006, 2008, the most recent is from 2013. None of these products, including this one that is a gene therapy, has been authorized yet, neither in the United States, nor in Europe. So the current treatment for alpha-1 antitrypsin deficiency is the one that you have been talking about all this time and that you know. Nothing new there. So what happens? What's the problem? You know, the problem, if you're interested in knowing these things, is that the development of medicines is slow and expensive, and you start with something like 10,000 compounds, and then you end up with one. And this takes years and years. Actually, how expensive it is, the industry quotes 2 to 4 billion of United States dollars. It's difficult to know because actually industry does not disclose their costs to us. So they can say more or less what they want. In any case, it's certainly expensive because you have to synthesize the compounds, you have to try them in animal models, you have to develop, to manufacture them. You have to be sure they are of good quality, particularly if they are biological treatments, for example. Not to speak about gene therapy or stem cells, something that is very complicated and needs to be standardized. Where do you take these cells? What do they do? You have different stem cells. Are they always the same? You know, do you put them in vitro with something and they develop into something else? How do you know that the batch A is the same as the batch B when you give the product, and so on? So there's a lot of work around here. And there is a lot of work in testing the treatments. And this is where it phases from phase 1 to phase 2 to phase 3. And then there is all the regulatory procedures, so where we intervene and we ask to the companies for more data and how this works and how that works and so on. So basically, the medicines reach the patients only here after all these years of history. So where are the problems? Why are we lost in translation here? So do we know enough of these diseases to get good treatments? Are we looking at the right disease? You know, also you know very well enough for antitrypsin, not everybody is the same. Some people will worsen faster, some people will worsen slower, some people will have certain type of symptoms, and people will have other type of symptoms. So we really need to, I need to go back. We really need to know the disease very well in order to provide people with the best treatment for them. Not for the group, but for the single persons. So again, do we study the right patients then in the trials? Because if they are the ones who will not exactly benefit from that treatment that we are proposing, because they are slightly different, how do we know if these trials were good or not, if this medicine will work or not? So, do we know enough? This is a bit complicated, but bear with me. Almost every disease is actually an interaction between some inherited characteristics, so some genetics, and some environmental factors. Some diseases are more based on environmental factors. Some, for example, diseases are based only on environmental factors. If you think at an attack of anthrax, anthrax basically makes everybody who gets in contact with it sick, so 100%. So it's purely environmental, no genetic factor. But if you think of Ebola, for example, that we all heard about last summer, not everybody who got in contact with the disease got sick. So there is always a mix of inherited characteristics and environmental factors in diseases. For alpha-1 antitrypsin deficiency, for example, we know that for the lung emphysema, the role of environmental factor, namely smoking, is very important, while probably, for example, for the liver disease, the inherited characteristic plays a bigger role than external factors. So, from this, actually, we have the base of how our diseases are born, are generated, and then we go to the cell, to the tissue, to what happens to our body in general. So, and then we go to the symptoms. How do we experience this? And then we go to how much impact this has on our family, on the society, and so on. So, if we focus on the first part, you know, we know a lot of alpha-1 antitrypsin deficiency because we are lucky enough to know where the genetic defect is, and for many diseases, believe me, we don't know. If you think about idiopathic pulmonary fibrosis, where people die in 1 to 2 years, they don't have any clue of whether there is a genetic defect or not. We know what this means. We know that there is a gene that has mutations, and this induces a protein that is either not working well or not enough, and so on. And then this can give us the possibility to build gene therapy and other approaches that then can actually act on the genetic. We also know how the deficiency acts in the body, so we know what it does to the liver and we know what it does to the lungs. And this also gives us the possibility of acting on these things. Actually, there have been some studies, very preliminary, either in vitro, in cells, or in animal models, where they show that some products can, for example, impede the polymerization of the Z alpha-1 antitrypsin, or, and the replacement therapy per se, in theory, even though it doesn't always work, in theory has a strong pharmacological concept, because if you think, I am missing something, I give it, it should work. But then it doesn't really work all the times. And then we know the clinical manifestations of the disease and what the disease does to the organs, the cirrhosis and the emphysema. And this is something where stem cell and alveolar regeneration are being studied. But how do we do? I mean, we don't have any of these treatments yet, although some of them are very promising. The only one that we have and that we are discussing all the time is replacement therapy. And I think it's also time, I know the Alpha-1 Foundation is involved in gene therapy, and I think it's also the time that we start actually thinking, as patients and as everybody, to basically how we foster the development of all the other possibilities that we might have to cure this disease, instead of just replacing what is missing, but curing it. This is an example, I hope it's not too difficult, but this is an example of how they did in cystic fibrosis. In cystic fibrosis, they developed this product, Calideco, and it was an association, in association between the industry and the patient association, actually. They found that there were some molecules that can basically modify the protein on the cell membrane, the protein that doesn't work in cystic fibrosis. And this was the first time that something like this was done, because until now also cystic fibrosis was treated in a similar way to alpha-1 antitrypsin deficiency, so with symptomatic treatments, things that make the lungs work better, and or antibiotics. But when there is a product that really works, so that really modifies the disease, you can see it. This is the lung function, and you can see this is the placebo group, and the patients that were treated really had a great improvement. And when something really works, everything goes fast, because basically from the discovery of this product to the approval, so to the moment when patients could have the product, only a very few years passed. However, this is not always the case. So if we go to our scheme that we had before, we realize that a lot of problems that we have in developing the products is that we test them on cells and or animal models. But does this really mean that they will work in humans? No. I made some examples for the lung diseases, and actually we use different models for that. Cigarette smoking, or some enzymes that degrade the lung, like elastase does when there is no alpha-1 antitrypsin in the body. They are all quite interesting models, and if you discuss with pharmaceuticals, or if you discuss with your investigators and they say, this worked in animal models, it's interesting to know what this means. These models damage the lungs, for example, the same way the deficiency of alpha-1 antitrypsin does. But they sort of reproduce some characteristics of the disease, but not all of us. I will go fast on this. Just to show you an example, that was basically a treatment that was supposed to regenerate the alveoli. And you see, this is the elastase model. So this is a rat that got elastase. So the lungs of the rat were destroyed. And actually, we had this product that was supposed, this is the normal lung. And we had this product that was supposed, look at this, to regenerate it. Wow, looks a little bit the same. However, we don't know if the cells that were, this is quite a complex architecture with cells that make you breathe. And we actually don't know if this was exactly the same type of cells that you have here in normal condition. It was a nice animal model, good results. The product was tested in patients. These are the results of the CT scan lung density. These are the results of the lung function. No change at all. So this is another example of the fact that medicine development can be difficult, and we need to keep in mind a lot of things. I wanted to show you these things because even if you are at the receiving side of treatments, I think it's very important for you to understand how this happens and where you can help, where you can understand, and where you have maybe just to be patient, not patients, unfortunately. So we also have problems sometimes in knowing, do our trials, clinical trials, fail because the product is bad, or do they fail because the trial is bad? So we don't know. Many, many times we don't know. Now, for giving you some lesson in epidemiology, we measure things that are, we use these things a little bit interchangeably. Outcome is what the trial is measuring. So I have emphysema, the trial is measuring lung function. But there are a lot of different ways of measuring lung function. For example, spirometry. Spirometry is an endpoint. Now, ideally, any trial would have the hardest endpoint possible. So how people function and how people live. So mortality. However, if you want to show a reduction of 40% in mortality in 5 years, you would need 684 alpha-1 antitrypsin patients with this FEV1 over a 2-year period. So it probably might be doable. However, it might not be enough. They did similar, yeah, expensive. They did similar calculations for COPD, and they realized when the trial was finished that was not still enough numbers for detecting an effect on mortality. So they studied like 30,000 people, and that was not enough yet. So these are paper calculations. So that's very difficult. That is to say that it's very difficult to have trials that can show an effect on something that is as important as mortality. So that means that whatever else we use, which is lung function, which is CT scan, is a surrogate endpoint, which is validated usually. So somebody took the burden of showing that it has some relation with mortality or not, because some of these have been validated in the 60s on 40 patients. But, you know, it's always to be remembered that these endpoints are variable, and, you know, they are not always the same. And depending on the population that you're studying, you might need different ones. This was something I presented at the last European Respiratory Society, and it's basically showing that this is not on alpha-1, this is on idiopathic pulmonary fibrosis that I was mentioning before. It's super deadly. It's like people die in 2 years, and there's nothing to do. And look at what they did. In all the trials that I've done, most of the trials failed until very recently. And when you look at the endpoints, so what they were measuring, they were all the time something different. Maybe they were the same outcome. So like a 6-minute walking test measure exercises, but sometimes was the change, sometimes was the changes in the lowest saturation during the walking test, sometimes was the more than 20% of increase. When you do trials like this in rare diseases, you end up with results that you cannot compare. So this means that actually it's very difficult to establish if treatments work or not when you cannot compare trials. So this is just a little bit of the landscape of what we have for alpha-1 antitrypsin deficiency, where we use FEV1, CT scan density. CT scan density is quite a good parameter for lungs because it's actually the one that goes closest to the lung structure. So it measures, you know, as close as possible, since we cannot take a piece of the lung from everybody each month to see it is improving or not. This is as close as you can get in vivo. The other tests are, of course, useful for many other things, but they have limitations. Again, depending on where you use them, on for what. And, you know, okay, just go on. So, this variety applies also to the way we design the trial. So for our long-treated patients, how often are the dosing similar? How do we compare trials of replacement therapy with augmentation therapy if sometimes patients got it once a week or sometimes got a higher dose or a lower dose? How do we know if we put all this data together? Because what we need here is numbers. The more people, the easier it is to draw conclusions that are mathematically significant. So how we do that if we cannot put the data together? So, for example, ignore the first one, but for example, as we mentioned, there are people that decline slowly, people that decline faster. How do we know if our patients that benefit from this treatment are the ones that decline slower or faster? If we don't select them this way, if we don't study them in this way before they enter the trial, and then we select the people who are most likely to respond from that specific treatment. Let's start from there. Then for the ones who don't respond, we can find something else. So, also, in many trials, we didn't notice significant changes at the CT scan. But when we think of using a treatment to think about this regeneration treatment or something that could really change the history, how long an observation is long enough? How long does a lung take or a liver takes to regenerate before we can see something that we can detect? Because like I said, we cannot take the organ out and look it under the microscope and put it back. We have to use surrogates, okay? So I will try to speed up, otherwise I will keep you here until the afternoon. So so after this, there is a lot of scientific questions. I put more questions than answers, but I do have some answers if you want. We can discuss it in the discussion. I will briefly go through how we authorize medicines. We are the European Union, and actually we are 28 member states now, and we do what is called regulatory science. So it is the science that is used to assess the quality, the safety, and the efficacy of human and veterinary medicines throughout all their development, but also when they are already authorized, when they are already used by the population. So in this way, we are basically here, a little bit in the middle, in between the industry that produces, the people who give the money, the academia who produce new molecules, the patients, and so on. So how do we do that? I've shown a little bit already before, and you probably are familiar already to the different phases of the clinical trials. Phase 1, that usually is on healthy people. It's more for safety and toxicology. Sometimes it's already on patients. Then we have phase 2, this is what we call therapeutic exploratory, so does the product work or not in a small group of people, and then the confirmatory is phase 3. And then at the European Medicine Agency, what we do is basically we authorize medicines centrally for the whole Europe. We also provide to the industry advice on how to develop these medicines through the whole phases, and we also provide orphan designation for the medicines for rare diseases, and so on. So, this is my committee. The Committee for Orphan Medicinal Products is composed by a member of each of 28 member states of the European Union. We have 3 members that represent patients' organizations. They are all patients, free patients, and they have full voting rights. And actually, that's how we make the decisions. Well, not with our hands raised, this was a joke, but we actually close, for all decisions we make, we close like 30 to 40 people in a room between 8 a.m. and 20 p.m. for 3 to 5 days, and then we go through a lot of medicines for all different diseases, and then we decide on each of them. It's quite a lot of work. What we do for the orphan medicines, we decide whether medicine qualifies to be a medicine for a rare disease. And we have a lot of inputs from the patients, particularly when we have to decide if a medicine brings an advantage, for example, in term of a new formulation that is basically making the life of patients easier, or a different administration route, for example, different dosing, like, is it an advantage that is big enough to have something orally versus IV when you have IV once a week versus oral 2 times a day. We have to make decisions on this to say this is a medicine that brings an advantage, so we can consider it orphan, which gives them rights to asking incentives and other things, and also gives them advantage to be reimbursed by the health technology assessment bodies, the ones who make the price for the countries or not. So these are important things, actually. So, what the orphan status does, like I said, companies ask for the orphan status because it gives them incentives. First, it helps them to develop the products because they can ask for European national funds, and then it gives to them 10 years of market exclusivity. It is important to notice that an orphan status in Europe can be granted to companies, to private citizens, to patients' organizations, to academia, to everybody. So whoever has a product that thinks it can work and has some proof of concept, so have some preliminary data on cells and in animals, can apply for that. This is our committee, so I will skip it. We have a patient and consumers working party that meets 4 times a year at the agency and gives inputs in a different number of areas, including safety of medicines, including transparency and communication, and it's involved in the scientific discussion and in the scientific decisions. And this is briefly, I will try to make this short, how we assess the medicines. When we assess the medicine, we have to decide on this. What's the benefit-risk balance? The benefit-risk balance means that for that specific population of people that need to be treated with that specific disease, with that specific age range, the benefits are higher than the risks in giving them medicine. That's as simple as this, but it's not simple at all. It means to go through a lot of material from the very early studies in cells, from all the studies on toxicology, huge studies on toxicology, from all the manufacturing, what I was mentioning before, the quality of the product. Is it made in a good way? Is it contaminated? Is it not contaminated? Is it good? And so on. So it's mainly about assessing the quality, the safety, and the efficacy. I will just go fast to this. So, is everything equal, or is some more important? For example, if you think at the combination of piperacin and artemisinin for the treatment of malaria, it treats the malaria of the Southeast Asia. Well, there's not a lot, and it gives some important cardiovascular effect. So there's not a lot of this in Europe. So while this has an important benefit risk in the Southeast of Asia, where the malaria of this type is endemic, so it would have really a good benefit risk there, it will not have the same benefit risk in Europe, because I can think that for one of my patients I have other alternatives, and it's very unlikely that they have this type of malaria, and the cardiovascular effects will not be worth the risk. These are all very important considerations. I can skip. And this is important to remember, because actually I also wanted to reassure you that actually usually we do not have major safety problems when we develop medicines. In fact, most of the medicines that do not reach the market fail because they do not demonstrate efficacy rather than because they kill people. So you see, safety is only the minority, but both in phase 2, the one I said first in patients, and phase 3, most of the drugs fail because they are not good enough. Now, when we look at what the patient think of benefit risk, we usually realize that us as regulators, the people who make this decision, and the clinicians, we have the tendency to basically think that patients, to try to keep the risk as low as possible. But patients, and there is a friend of mine studying these things, asking for patients' preference. I was discussing this with Shane before. She's working on multiple sclerosis, and she's creating questionnaires such as, do you prefer to have 10 years without a relapse of multiple sclerosis with some risks of your medicines that are A, B, C, D? Or do you prefer less risks and, let's say, the risk of a relapse every 2 years? And patients basically build scores on this and help to create the trials and help to decide on the benefit risk of medicine based on this. So I think this is something maybe important. It sounds more complicated than what it is, but I think it's important also for the Patients Association to keep in mind that if you basically decide to work with the right people, you can do this type of reasoning as well for your own disease. Because patients, as you look from this, are usually ready to accept more risk than what parents are and what physicians are. I've almost finished. So what comes out of the assessment is that we have a medicine that is licensed for a specific therapeutic indication within that population. So it's not for everybody. It depends on the persons that are studied in the trial. So the age range will be that one. The severity will be that one. And it becomes difficult then to use this medicine in patients that are older, younger, that have not been studied. So it's very important that this is also to be kept in mind. And this has to do with what we call comparative effectiveness. So while efficacy is what you demonstrate with the trials, effectiveness is what you demonstrate in real life. So what does the product when it's out there, when you're using it with all the other treatments that you are already using? Because you will not be your whole life in a trial. You will be most of your life in normal life. So you will be taking other treatments. You will be taking the treatment in a slightly different way from when you were in the trials. You will be a slightly different person from the one who was in the trial, maybe bigger, maybe slimmer, whatever, okay? So this is very important because there are ways to study this. I'm finishing because they kill me otherwise. It's not only us who make decisions There's a very important part here, and I'm sorry that I have to go through this very fast, but we can discuss this in the questions if we have time for the questions. There are the people who have to pay for the medicines. The health technology assessment, the one that will look at this, comparative effectiveness. Oh, these guys at the European Medicine Agency have authorized this medicine because they had some good results versus placebo. But there is not only placebo, these people are treated with other things. Is this medicine really good enough that my country should be paid for this? And there's a lot of discussion about this. Okay, so we make a decision on the licensing, but there are a lot of different HTA decisions. And I just skip this, and I want to go to some actions for you, and I will stop it here. I think you can stimulate the companies to an early dialogue with regulators on the innovative products, the one I was mentioning, the ones that are not just the replacement therapy, but gene therapy, molecules that can unfold the protein that is folded in the liver and gives the liver toxicity and fibrosis. You can help and promote the study of phenotypes. Oops. Oh, where's that? And different forms of the disease, and working together with the registries, for example. And you can participate in discussion and the creation of endpoints in the way I was saying before. Do you prefer, this versus that? Okay? Please also stimulate the scientific community in the consistency in clinical trials and real-life studies for comparative effectiveness. There are a lot of studies that can be done once the products are already on the market to study, and this helps also for reimbursement. And then, well, participate in development of treatments for COPD in general, and I will skip the rest and close here, and thank you for your attention, and sorry for being long.